Welcome back, everyone. We're happy to welcome Clene Inc. It trades on the NASDAQ under the symbol CLNN. As a late clinical stage biopharmaceutical company focused on improving mitochondrial health and protecting neuronal function to treat neurodegenerative diseases. Please welcome its CFO, Morgan Brown, and President CEO, Rob Etherington. Welcome, gentlemen.
Thank you.
All right, the floor is yours. Call me back when you're ready for questions.
Yep, sounds good. Thank you, Anna. And good afternoon, everybody. We'd like to welcome everyone to the Clene Inc. presentation at this Emerging Growth Conference. We're excited about the opportunity to present our story to the investors in this virtual audience. By way of introduction, my name is Morgan Brown, and I'm the Chief Financial Officer of the company. And joining me on this webcast is Rob Etherington, Clene CEO and President. He and I will tag team the presentation and Q&A session of this meeting. Both he and I are dialing in from our headquarters in sunny Salt Lake City. And additionally, we have proprietary manufacturing facilities in Maryland. As a reminder, Clene Inc. is a NASDAQ-listed company trading under the symbol CLNN. Due to being public, I need to remind everyone that we'll make several forward-looking statements in our presentation today, and we have no responsibility to update these statements in the future. Let's now jump into our presentation. Clene is a biopharmaceutical company focused on transforming the treatment of neurodegenerative disorders by restoring and protecting neurological function. In this slide and subsequent slides, we'll share specifics on how we are doing this in a very unique way. I'd like now to turn the presentation to Rob, who will present in more detail our approach to treating neurodegenerative diseases. Rob?
Thank you, Morgan. So the WHO has predicted that neurodegenerative diseases are one of our true unsolved problems. And I hear now almost every day of a new MS, ALS, or Parkinson's diagnosis. And there's many spaces that we could approach that we have not yet done. But I want to review with you why we have built the clinical case for neuroprotection in neurodegenerative disease. Everything I'm about to talk about is organic to Clene. We built it here. We organically grew this here. And we have 150 plus patents and numerous trade secrets to support what we're doing with this asset that I'm holding up in my hand, an extensive conversation which we'll now review about our approach with CNM-Au8 in neurodegenerative diseases. Now, all such diseases, again, I've mentioned specifically our key focus, ALS, amyotrophic lateral sclerosis. Some of you may know this as Lou Gehrig's disease. The famous baseball player in the 1930s famously, if you will, gave his name to this disease because it was the first time that most in the American public caught word of this horrible, devastating condition. Outside America, it's often called motor neuron disease. And if we go to multiple different countries, there tends to be athletes that are really quite a focal point of the disease. Now, for reasons unknown, athletes have a predisposition to get the disease, but it is certainly not only athletes. It's men and women of all different types and ages. And it remains most devastating with neuronal death. Neuronal death that then causes a compromise of function. It compromises a compromise of any muscular, rather it composes a compromise of any muscular movement that controls how we work, how we eat, how we think, in MS anyway, and in ALS, how we breathe, how we chew, how we talk, how we swallow. In other words, all neurodegenerative diseases have an impact on our central nervous system because dying neurons exhibit hallmarks that converge on mitochondrial dysfunction and nicotinamide adenine dinucleotide pathway deficits. That's a mouthful. Let's just refer to it as NAD+. And what you can think about it is the way your neurons use energy to drive their function to move muscular movement throughout the muscles of the body. And again, we're not only talking the way we move and walk and lift things, but the way we breathe, the way we swallow, the way we talk. So let's take a moment now and look at a video that will explain this for the next 2 minutes. Then I'll return to the slides. Operator, please cue the video.
In the human brain, Specific populations of neurons must utilize large amounts of energy in order to function. These populations are associated with neurodegenerative diseases such as Parkinson's disease and amyotrophic lateral sclerosis. In neurodegenerative diseases, neurons lack sufficient energy to perform their normal functions. Aging-related genetic and environmental factors combine to cause these neurons to undergo an energetic crisis, resulting in toxic protein aggregation and accumulation of harmful free radicals, leading to neuronal death. CNM-Au8 is a suspension of gold nanocrystals that improves mitochondrial function and reduces oxidative stress and lowers levels of misfolded proteins. Clene Nanomedicine discovered that gold has extraordinary catalytic therapeutic activities when it is engineered as clean-surfaced faceted nanocrystals. The surfaces of gold nanocrystals, among other capabilities, are able to rapidly catalyze NAD, a key energy metabolite. This catalytic reaction drives the production of adenosine triphosphate, ATP, the cellular currency of energy. Here at the surface, harmful free radicals are also rapidly converted into non-reactive species, such as water and oxygen. By restoring NAD+ supply and improving mitochondrial health, CNM-Au8 protects neurons from disease and death, helping them survive and function. CNM-Au8 has the potential to transform the treatment of neurodegenerative diseases by restoring and protecting neurological function. CNM-Au8 is an investigational compound that has not been approved for use and clinical benefit has not yet been established. The safety and efficacy of this compound has not yet been determined.
Okay, thank you for taking a moment to look at that video. I think it adds some good color and character to what we're talking about. Our asset, CNM-Au8, which again I'm holding here in my hands, is all around enabling surface catalysis to improve mitochondrial function. And so patients are drinking this. Let's be clear. Patients are drinking this every morning in these clinical studies and in our compassionate use early access protocols. And when they do so, over time, there is an improvement of mitochondrial function leading to neuron survival and function. Now, if I had a water glass or our asset and I drank either, I would not be able to tell the difference. Our drug, CNM-Au8, is remarkably easy to drink and has no taste profile. And so by consequence, many clinicians tell us this is among the easiest asset they've ever administered in a clinical study. So now let's talk about those clinical studies. And I'm going to run through a bunch of data. So thank you for staying with me. All of this website content is actually the presentation I'm showing you, these slides, that is, are on our website presently. Now, the first studies we did were called Repair. We used a very high-powered MRI machine, 7 Tesla system, to really look into the brain of patients and understand blood-brain penetration and target engagement, and to prove that the energy metabolite NAD+ and the energy metabolite ATP were either improving or being normalized, as we stated earlier in preclinical studies, and as we stated earlier in this presentation, needed to occur. And indeed, we saw this in a statistically significant way in early Parkinson's and stable relapsing MS patients. Again, the essential energy metabolite that you and I rely upon to enable our central nervous system to work properly was improved. And what that leads to now is the clinical data in ALS first, where I will focus, and then MS. But before I get there, I want to talk about our safety. We have over 600 collective years of subject exposure, now nearing 650, in fact, without any identified safety signals across either of ALS, MS, or Parkinson's. So again, Lou Gehrig's disease, ALS is the proper term now, multiple sclerosis or Parkinson's, we have seen not a single serious adverse event related to drug, CNM-Au8, that has caused any major showstoppers. And in our world, pharmaceutical drug land, those are called SAEs. Now, we have some side effects. All drugs do. Ours are mild to moderate, rarely cause any patient drama at all. And they tend to be entirely GI or headache when seen, but again, relatively rare. So if you were to talk to clinicians involved in our program, they would again tell you, as I've mentioned, that our drug is very easy to administer and to tolerate. Now I'm going to specifically focus on ALS. We've completed 3 projects in ALS that have had a fair amount of now breadth and depth. The Rescue Project was a phase 2 clinical program of 45 subjects, originally in double-blind, placebo-controlled studies for 9 months, with now many years of follow-up. In fact, we're now nearing data we'll be releasing later on this year that is up to 4 years of total administration. The Healy study was 6 months in duration and double-blind, placebo-controlled, and we have over 133 weeks, so nearing 3 years, and a real-world experience in compassionate use where our drug was offered to patients who did not qualify for either of Rescue or Healy, and they were too sick, in other words. They did not meet the inclusion criteria. And we have those patients numbering 256, and they've been on drug for over 4 years. You see a lot of check marks here where we've achieved something. Let's talk about that in granularity next. Both Rescue and Healy showed a statistically significant improvement on the exploratory endpoint of clinical worsening. So clinical worsening in ALS is defined as whether or not I die, which is an obvious form of clinical worsening, whether or not I need breathing support, either mechanically through a tracheostomy that's cut into my neck and a machine breathes for me, or whether I need assisted breathing through BiPAP or CPAP during the course of my day or night. And then finally, whether or not I have lost the ability to swallow food or water entirely, so I need a G-tube into my stomach, and all of my food and water is administered by a gastrostomy tube. So those are, as you can certainly see, devastating impacts for the families and especially for the people who struggle every day with ALS. And as you see here in green, those are the number of clinical events of constituting clinical worsening that occurred on AU8 patients. And in gray or black, probably it looks like to you, the dotted line is the number of clinical events for placebo, and both are statistical significant differences. Continuing, most importantly, is survival benefits. So we see pretty consistently a 70 to 90% risk reduction, depending upon how we look at this across our clinical programs, including their open labels. The key thing I want to focus on is here in the Healy program, we saw survival as the secondary endpoint in the 6-month double-blind placebo-controlled study. So let's tackle these projects now, these clinical programs, one by one. Rescue first. Rescue in Australia was early patients. They were only 4 months diagnosed. We enrolled both bulbar and limb onset subjects. So ALS either is bulbar onset, which is kind of neck up compromise, or limb onset, which I noticed through challenges in controlling the muscles of my hands and arms and legs. We enrolled both patients. We missed the primary endpoint because these patients were so early in their disease progress that the 30% of patients that were bulbar never progressed to limb involvement. We did see, as a secondary improvement, Munich's change significantly on the limb side, but it didn't count because the pre-specified primary endpoint was both subjects, bulbars and limbs, at the primary endpoint. However, we learned a lot about this study that has taught us ever since, because we saw significant ALSFRS at 6-point and higher. We saw significant in the 9-month program with both subjects. I'm talking now both subjects, bulbar and limb. We saw quality of life changes, and we saw clinical worsening, as I've just explained. That clinical worsening continues to sustain during the open label extension. Even still, with the subjects that choose on placebo, choose to go on active drug as fast as they can, which is what you and I would want if we had ALS, to go to active drug that might help us stave off the disease progress as fast as we can. Even those subjects, you see here a flatlining of their events. Once they moved on to AU8, they stopped declining, but they never really caught up for the 9 months originally lost. And we always keep a statistically significant improvement of clinical worsening for those subjects. So we also saw as we followed these patients over time, a survival benefit that gave us, from our disclosed data on rank preserving, about a 19.3-month survival benefit. In other words, patients on drug were estimated to have lived in the rescue study 19.3 months longer through rank preserving survival time failure modeling, longer than those who were on placebo. And that is consistent as well with the long-term survival benefits against matched PROACT placebo controls. What PROACT is it's more than 12,000 patient records from multiple clinical studies that have been gathered to help assess clinical studies in ALS, where everybody is declining historically in clinical studies, but they also want to go to active drug as fast as possible. So as soon as we give them permission, we then have to start comparing their situation against matched placebo controls in the historical database of trials. That's called PROACT, and we see here statistically significant improvement over time. Now let's jump to the Harvard Healey program. This is the most important ALS study probably ever attempted. It's named the Healey ALS trial platform trial because of the very generous donation of an ALS patient by the name of Sean Healey. And the Harvard ALS Clinic was renamed in his honor, and a platform study was commenced. A platform study is where you take multiple drugs and you study them at the same time with the same primary endpoint and the same secondary endpoint to see if you can accelerate drug development in ALS. And so these regimens are: A was the pharma company based in Europe called UCB, B was Biohaven, C was Clene, D was the private company Prelenia. Also, Healey has just concluded earlier this year, regimen E for Selos. In all the cases, the 5 completed drug companies so far have missed the primary endpoint, change in ALS-FRS slope adjusted by mortality. Clene has achieved a survival benefit in the double-blind placebo program and clinical worsening in exploratories and neurofilament achievement. So let's talk about these one by one. So first, here is the survival benefit. I've already showed you this data: 90% risk of reduction of death, 90% reduced risk of death with AU8 30 milligram. That was significant whether I looked at the drug versus all of the placebo, in other words, shared placebo across the 4 regimens, or whether I look within our own Clene specific regimen. We also saw a delayed time to clinical worsening that was significant as the exploratory, which I showed you earlier. And we also showed a neurofilament biomarker change. So some of you might not be familiar with neurofilament. It's very relevant because as the disease ALS attacks the neuron, we can measure in the blood the, shall we call it, the debris field that is left behind as the neuron is so compromised. And it's called neurofilament light. So I can measure evidence of a neuron attack in the bloodstream. And this is very relevant because Biogen received an approval for their drug called Qalsody, generic name tofersen, about 2 years ago, based upon neurofilament alone. Biogen, in their primary study, missed the primary, secondary, and exploratory endpoint, yet the agency saw their neurofilament benefit in SOD1 homogeneous disease. These are ALS patients that are missing a specific gene. It's a very small piece of the ALS population, but a very important development for those patients who have this gene in a devastating way has run through their families. They benefited immensely, and the FDA approved the drug. So what we see here is a statistically significant improvement at a p-value of 0.04 of a neurofilament improvement. And if we look at the patients who have higher baseline that they started the study with, there's an even more material improvement. And as we look at this further and look at these patients in open label, which is green, versus the placebo folks in gray, it becomes a higher p-value and a 70% improvement even on the double blind. And we can also correlate that improvement to clinical benefit by accounting for the same clinical worsening I counted earlier, which is whether I die, whether I need assisted breathing because my lungs can't power my breathing, so a machine must do it for me in some measure, or whether or not I need a G-tube. Active patients in green, the placebo in gray during the double-blind period, and we see quite appreciable number of events if you're on placebo versus active And the more your neurofilament rises, the worse it is. So this is very relevant because it also gives us an understanding of why the survival benefit was seen in the Healy program. And as we see here, the green is active, gray is placebo, the same statistical significance you saw in the rescue study. And we also see, similarly, the ring-preserving crossover adjusted survival that is quite materially different and highly significant for those patients on drug versus original placebo. Now, third, we have this extensive compassionate use program, 256 valuable subjects, of which we have 220 with baseline data that we can measure. And in that measuring, we find that against PROAC and the natural history data set, we also have strong survival with hazard ratios of 0.4 and 0.3. In other words, a 60 or 70% survival benefit with reduced risk of death that was highly significant. And so given this, the National Institute of Health has just given Clene, bless the government of America, to help us pay for this, to give access to many more patients, $45 million that we just announced. We've started to receive now last month, and we now are working to enroll an additional number of patients in our EAP. So now I want to change gears to MS for a minute. The same drug, CMAU8 30 milligram, has been studied in the Visionary study. And in this case, we're looking at MS patients who have optic neuritis. So this is about 50% of the MS population. These are MS patients that they're not dying anymore because we have great drugs that treat MS that stop the immune attack and prevent MS lesions from occurring because there's a tamping down of my immune system that is causing an attack on my own neurons. These drugs work fantastically to stop MS attacks, but we still have many patients that have functional decline. They can't see, they can't move, they can't walk, they can't think because of the MS compromise. And so those are exactly the MS endpoints that we were looking at. The primary was vision, the secondary was fine motor or cognition with single digit, walk, 25-foot walk, or again, vision, looking at this as a composite endpoint. And the study was only half enrolled because COVID impacted it immensely. Patients on DMTs, tamping down my immune system, did not want to go in MS clinical studies where there was a line of COVID patients outside the clinic. And so by consequence, when we enrolled this study, which was 2020 and 21, we started it just before, in the second half of 2019, before any of us had heard of COVID, and got a fair number enrolled, but then our enrollment was stopped flat. We opened this study expecting we'd see nothing. And so before we opened it, we asked the agency what we should do in a half-enrolled study, and we pre-specified a statistical threshold set at 0.1, and we were stunned to find that we got a P05 on the primary endpoint of vision improvement, which was 3 extra letters that over the year patients could read. Unprecedented improvement. We've not seen before in MS disease an improvement in the optic nerve and the ability of the eye to read and to therefore see things. We also saw the secondary had a P of 0.2 when we looked at the composite endpoint of cognition, vision, fine motor movement with my fingers and thumb, and walk. And even more interesting, we just announced in Denver at the American Academy of Neurology that the longer you're on our drug, the more you improve your vision, up to 8 letters on this eye chart. And that patients on placebo that move to active drug, which is this graph on the right, over the 2 years of follow-up, they started replicating the same vision improvement. Why did this occur? Because we are using biomarkers, improving the way that patients are seen by improved amplitude and latency as measured by visual evoked potentials, a very sophisticated technique that understands the signal strength and speed from the eye to the visual cortex. This is data suggesting that there's evidence of remyelination and improved axonal integrity. So the reason the eye is seen better is because the way the eye sees and the way the neurons are processing information is improved. Now, that's not surprising because the secondary endpoint cognition also saw improvement that continued not just during double-blind 48 weeks, but in 2 years of follow-up. And again, we see concordant data that the placebo folks, once they move to active drug, they're also seeing a cognition improvement. Why did that occur? Because we see the same U-shaped curve here with the bottom being placebo and the top being active, that when patients are on drug, their whole brain white matter is preserved because we're seeing a demonstrated MS lesion reduction and promoted myelination, remyelination, that is, within the brain. And the placebo subjects, once they convert to active drug, which is the second 2 years, they improve. So my last slide, what's next? We are, as we've announced in our Q to the SEC and to other investors, including right now with you, we are preparing now a briefing book that we'll be sending to the agencies shortly to talk about the possibility of accelerated NDA accelerated approval. Now, we had already had one conversation with the agency late last year. We announced this to the public markets in December. You will see that when we did that, the public markets penalized us because that first meeting with the agency said, we're not going to approve you today at this time because we need more. We need you to come back to us and explain more about biomarkers, explain more about your mechanism, explain more how these correlate together, explain more about your phase 3 plan, because that's part and parcel of an accelerated approval. And we need you to talk about survival in greater detail and correlate it all together. So that's what we've been working on the last 4-5 months, getting more biomarker data, more mechanism understanding prepared for the agency, correlating it together and getting thought leader community prepared for the global launch of a phase 3 later on the second half of this year. And we'll have those conversations with the agency in the Q3, as we've already announced. Now, in the case of MS, we are waiting for the wrap up of Repair MS in progressive patients. These are patients who have MS but progress independent of DMT. And then we'll be hosting end of phase 2 meeting with the FDA and possibly commencing with a phase 3 study in MS for what is truly the market need, patients that are progressing independent of relapse. So a fair amount of catalysts that are coming up in the second half and in the first half of 2025. Let's now turn to our last slide and have Morgan talk about our plan and approach for those catalysts.
Thanks, Rob. As you guys can see, a lot of exciting stuff's happening here at Clene, a lot of terrific data that we've released, and look forward to those conversations later this year with the FDA about a potential of a path forward for accelerated approval. As Rob mentioned, cash runway is an important component of any biotech company, and you can see on this slide that we finished the first quarter with about $28 million of cash. And if you look back historically over the last several quarters, we burn on average $7 million a quarter. So based on that, you can see that we've got cash that will take us into the first quarter of next year, with one small caveat. We do have some debt that right now, under the terms of the current facility, principal begins to be repaid in July of this year. We are having meetings with our debt provider and hoping that they will, negotiating with them to potentially defer further those principal payments and allow that facility to be interest only through the balance of this year, which will extend that cash runway into the first quarter of next year. Additionally, we continue to explore ways to bring in non-dilutive capital to the company. We have an ongoing licensing/business development initiative underway at the company, in which we're talking to several global as well as regional players about the potential of licensing either ALS or MS or both indications to a third party that will bring into the company some non-dilutive upfront capital, as well as help us in the funding of future phase 3 clinical trials. And the last thing I'll mention before we open this up to potential questions is that last year we did raise $40 million of capital with the likes of Vivo and Acuta and Special Situations, some great funds that are dedicated to this healthcare space, but they wanted to make sure that that could potentially have been our last financing. And so we issued them 2 tranches of warrants tied to regulatory milestones. The first tranche is tied to regulatory submission, which, as Rob mentioned, that could happen here in the second half of this year, and that would bring in another $55 million of capital. And the second tranche tied to regulatory approval, which could happen in the first half of next year, which would bring in $75 million of capital. So the combination of both of those tranches of warrants would bring an incremental $130 million to the company, which would extend runway, a significant period of time and allow us to hopefully commercialize ALS through an approval of the ALS indication. So with that, we will open it up to questions and answers. Anna, maybe we'll turn the time back to you. I think you may have some questions already in the queue. Maybe you can share those. And then as others have questions, we look forward to answering those as well.
Wonderful. Thank you for that thorough presentation. Yes. Now, Rob, I know you mentioned this in the presentation, but tell our listeners again the timeline of meeting with the FDA to discuss the accelerated approval pathway, including when a potential NDA would be submitted and when a potential AdCom and PDUFA data would be.
So we are preparing the final stages of a briefing book now. We've already told the marketplace in our filings that we'll be submitting this briefing material to the agency by the summer period. So that's now. It's 92 degrees here in Salt Lake City outside, so that categorically qualifies as summer. So within a few weeks, that will be submitted, and then the agency has a time clock where they review this. They accept it, review it, and then we'll be taking experts with us to these meetings. These meetings are also going to be held during what some of us would still constitute as summer, but in our briefing materials to the SEC, we've constituted this as the Q3. The agency will then take under consideration these 4 themes that I've just referenced. And again, for the viewers, I'll remind you, the design of our phase 3, which is a key part and parcel of a conditional accelerated approval. And by the way, that is, in Clene's case, going to be a time to event study. We're looking specifically at keeping people alive and surviving. That is our goal. That's the way our drug works, keeping them alive and surviving. And so time to event in pharmaceutical parlance means death. So survival is our goal. We're taking a mechanism of action discussion. How does CNM-Au8 work by oral administration? 3, the biomarker data, and I speak biomarkers plural. We've announced neurofilament, but there's other biomarkers that we've not yet announced, which we are telling the agency about, and we'll announce them at scientific meetings here in good time. And then 4th, the correlation of all this together to survival. So again, that discussion will be over the Q3 period, and then the agency will get back to us on their timeline, which will be Q3, maybe Q4. It's hard to sometimes predict. And then the goal is that we would then go to an advisory committee. If all of this works, we'd file an NDA, a new drug application that would go into the agency. There'd be an advisory committee, if you will, a hearing that would then enable a full comprehensive review of all of our data and safety, and that then could lead potentially to an accelerated approval conditional upon a successful phase 3 completion that we would be able to announce somewhere in the first half of next year.
Sounds like you have a busy summer. That sounds great. Thank you, Rob. And Morgan, remind the audience the amount of current cash, which is on hand, and the runway, what it will be with that cash balance. Also share some opportunities you're currently evaluating to strengthen your capital balance using both dilutive and non-dilutive sources.
Yeah, no, good question. And just to reiterate, we finished the first quarter with about $28 million of cash, Anna, and burn on average $7 million a quarter. So if you just do the quick math, that's 4 quarters of cash beyond, sort of the end of the first quarter of next year, which takes us to the end of the first quarter of 2025. Now, I did mention we do have some outstanding debt that we're right now in the process of negotiating with a lender to potentially delay that principal repayment, right, and extend the interest-only period. That's not finalized yet, but assuming we're successful, we should have capital through the first quarter of next year. As I mentioned earlier, we are exploring other ways to bring in non-dilutive capital, including potential licensing transactions with either global or regional pharma players to license our assets in MS and ALS indications. Once again, timeline is similar to the FDA discussions. We should know the outcome of those discussions and that diligence process by, call it, end of summer of this year.
And Rob, when do you anticipate initiating the P3 clinical trial in ALS? Has that trial already been approved by U.S. and European regulatory bodies? The answer is we've not announced publicly its approval process, so that's still in progress, but will soon be announced. And the key piece of this is we need to still fund that full program. It's an expensive initiative, and we want to make sure that it's fully funded effectively, or at least enough funded to commence. We hope to commence that by the end of the year based upon the successful completion of some mixture of the things that Morgan talked about, including
a possible partnership engagement with Clene from a big pharma company, including a catalyst-driven capital raise at the right point. We've purposely not done this yet to avoid diluting current investors, so we've been very sensitive to the present share price. And third, based upon if the agency concurs with our accelerated approval path, then that would obviously be a very material catalyst that would enable the funding of this program.
Thank you for that. And outside of CNM-Au8, are there other assets in your pipeline? And what indications are you targeting with those pipeline assets?
So CNM-Au8, we have a pipeline of discovery, to answer your first question, a pipeline of discovery. If you can think of a therapeutic elemental metal that has medicinal efficacy, we can prepare such an asset. However, knowing that this single asset at 30 milligrams now has multiple opportunity in not just PDMS and ALS, but in other rarer and orphan spaces, we have actually, to preserve investor capital and be wise stewards of that, we have focused this program on the progress that has the fastest road to commercialization market, which frankly are the neurodegenerative diseases we speak. But as soon as we make progress sufficient in that respect, we plan to initiate clinical development of these other assets in our pipeline.
We have a question from our viewer, Scott, wanting to know about when it comes to the vision and fine motor skills you're monitoring, after you get off the drug, how quickly does it deteriorate?
So that is a good question. It takes a moment to get up to full steady state for Clene, 3 to 6 months. And we're finding similarly that once you're off the drug, it takes 3 to 6 months to lose full effect. So that is the answer. About half of a year before one sees the deterioration that you had pre-drug return. That is a great question, though, that is still being scientifically understood and addressed. Because most of our patients have stayed on drug in the open label program. If they're on placebo, they converted to active drug, as I've just explained. And if they were on active drug, they wanted to stay. So we don't have full vision yet to how fast you lose full effect. But we are expecting from some patients' experience and from just the thesis of get to steady state, you're on drug, everything's fine. You stop taking drug, you lose drug levels. That's the thesis we have at the minute. What's also a key question, though, is do you envision combination therapy? Absolutely, we do. I didn't mention that at all. Our drug is taken on top of standard of care. So whatever the present patient has been prescribed by his physician, whether he's ALS or MS or Parkinson's, or she's ALS or MS or Parkinson's, they stay on that therapy. Our drug is not competitive in the liver with another drug, which is a big advantage. We basically have no drug-drug interactions, so we let the patient come to us on whatever drug they want, and we put our drug on combination. And to answer Scott's question, it is understood that patients will stay on our drug chronically. They'll take it as long as they feel the need.
Thank you for that. Robin asks if you're still on target for the first patient visit for the ACT EAP this month.
We are, yes.
Okay.
What Robin's asking about is the compassionate use program that's funded by the National Institutes of Health. We've had multiple requests. There's a long series of patients waiting. And in partnership with Columbia University and Synaptic Cure, we do expect first patient, first visit this month.
And John asks, do you plan to ask for accelerated drug approval?
We do. That's the entire thesis of the thesis with the agency, to show the biomarker data. Again, as I mentioned, to show mechanistically how the drug is working, to tie it to survival, and to show the correlations thereof, and to ask the agency if that's sufficient to have an accelerated approval conditional on a phase 3 that concludes.
Great. Thank you for clarifying. Harold says the $45 million, your government-assisted program, is that a grant? Can you explain that?
It is. So President Biden, in a great act of bipartisanship, let's count every victory in that respect right now, signed into law the Act for ALS, which gave $500 million. It was $100 million a year, beginning in 5 years. $100 million a year over 5 years, hence the 500 mil. And that 100 million is set aside for research and compassionate use. And we received so far the largest grant from that capital, which is the $45 million in question. It is a grant. It enables us to accommodate now 180 patients altogether in that compassionate use program. So we have 256 that I've already referenced that Clene is paid for out of our own capital. And this enables us to actually do another 180, including all the clinical work that the agency requires for that. So nearly doubling the number of patients we can help. And yes, it's a grant, meaning Clene didn't have to, pay any of the costs of this incremental number.
And if I could add to that, it's a 4-year grant. So that $45 million is not a number that we got in advance, right? As we incur expenses, we submit those for reimbursement and then are reimbursed for those expenses. And that $45 million will take us roughly, call it $10 million a year over the course of 4 years.
Perfect. Thank you for that clarification. Will says in May, your quarterly financial miss was significant. However, stock remained basically unchanged. So has this hindered in any way your ability to stay funded? And a follow-up, how far out are you currently funded?
Yeah, once again, when we talk about misses against analyst projections or otherwise, right, we're a pre-revenue, pre-commercial company. So I think that's somewhat irrelevant at this stage of our business's existence. I think the focus should be on how much cash are we burning on a quarter-to-quarter basis. And we've been very consistent of $7 million a quarter for the last 3 or 4 quarters. And as I mentioned earlier, right, we've got $28 million of cash at the end of the first quarter that if we're successful in delaying principal payments on some debt should last us through the first quarter of next year.
Thank you. Shalom asks if you can quickly summarize the status of the products you have in your pipeline, basically the state of testing and regulatory approval of them.
So CNM-Au8 is the main asset. That's the lead asset I've held up a couple times and that we're developing for ALS and for Parkinson's disease. And MS, and that is the asset that we're taking to the FDA later this year for the possibility of accelerated approval. That same asset, we're having an end of phase 2 meeting on multiple sclerosis later on this year as well. And then other assets in our program, as I noted earlier, are still in clinical development and will have future conversations with the FDA.
Perfect. Well, thank you, gentlemen, for this very thorough and meaningful presentation. Do you have any closing remarks for our viewers today?
Just to say that we're doing something very novel and unique. Nobody else has ever tried a nanotherapeutic. So whenever there's a break with convention, there's a breakthrough. And we think we've got that. What we're seeking here is that we can give the mitochondria that is enabling the energy that you and I rely upon to move and walk and talk and eat and chew and breathe and think, if I have MS, that we enable the central nervous system to do all the things that we take for granted, and that we enable that because of an oral administration of a drug that is super easy to take, and that we see a fair amount of clinical data to support the thesis that we've just laid out in the last 40 minutes. So thank you for taking the time to think about Clene, to listen to us, talk about our plan for neurodegenerative disease, and thank you too to our investors for supporting us in this mission. And we thank you for the time of this Emerging Growth Conference. And Anna, thanks for hosting.
Wonderful. Thank you both. We look forward to having you back with some updates in the future.
Thanks, Anna.
All right, everyone. Thank you, guys. We do have an update from Region Biopharma. It trades on the OTC Pink Sheets under the symbol RGBP. We learned that David Kos was placed. He had an adverse reaction to an antibiotic recently treating kidney stones. So he will be back in July. So we wish him well, and we'll be back very shortly. Thank you, guys.